Okay. So anybody has have like comments of their experience of what happened in lab on Tuesday?
We were most interested by the last try with just the water and then seeing the bubbles go up and trying to figure out why.
All right, so that is a really important point. In fact, I think we can start there. Let's start there. That is a really important thing. I'm glad you noticed that. I'm gonna, in preparation for doing this, I printed out. One person actually typed in their data, and I made a quick little graph of it so I could talk about this stuff more easily with you all. This was one person's data. Trial 6, the pure water is this green one. So mainly I want people to look at what would you expect if you ran the experiment with just pure distilled water in terms of bubbles?
Flat line, none.
None. In fact, it's kind of our control to make sure that there's no reaction if there's no substrate. And as we can see here, it's like, oh, something did happen. There was like a big change in the first 5 seconds, but then it was flat. Nothing changed. So first thing tells us, okay, something's going on we need to pay attention to, because if everything was as we expected, there would be no change when we added the distilled water, but there was a change. There was a big bloop in the beginning here. So what is probably happening? Why did this actually go up? Why did the oxygen evolved, so to speak, in this thing go up, even though there was no reaction?
We thought maybe it was just like air got pushed through, because it happened to me. I think that was my group. It happened immediately. Like as soon as we pushed it in once, we saw like an air bubble go up, and we were surprised by it, but that was kind of what we figured had probably happened.
Right, so think about this. You've got this little sealed thing here. You got one thing going out into the tube. We've got some stuff in there. And then the other side, you had that syringe. Five mils of H2O. So this is trial 6 here. So when you at time equals 0, you're injecting 5 mils of water down into here, right? What was in, even though it was invisible, what was in that flask before you injected the water?
Air.
Air. There's air. It's filled with air. You just don't see it. But if you take 5 mils of water, it has to push something else out of the way to get in there. So the air gets displaced. Where's that air going to go that's getting pushed out of the way as that 5 mils of water goes into the flask?
It goes up the barrette.
Yeah, it's going to get shoved up here and bloop, bloop, bloop. And you're going to see it as this 5 mil bubble going up as you displace 5 mils of air that was previously taking up space, minding its own business, just hanging out in the Erlenmeyer flask. And all of a sudden, this 5 mils of water gets shoved in here and 5 mils of air has to get pushed off someplace else. So that's important information that you are going to be able to use in your, both interpreting your data and in your discussion. The fact that we know that that first 5 seconds or so isn't real. there's like this weird bubble coming up. So the first 5 or 10 seconds, you probably don't want to trust. What about the bigger picture? Does it seem like there is a reaction with distilled water?
Nope.
No, because once you have that artifact, this is flat as a board. So this is actually telling us two things. One, it's telling us like, oh my God, there actually is an artifact that we need to pay attention to and not trust too much the initial readings here, because there's a weird, big, huge bubble that's getting shoved up into there. But then it's saying after that artifact is done, this is stable. So it's all good. So trial 6 is going to be important. It gives you information. It's saying, and what is the slope? What is the reaction rate here if the slope is just a horizontal line?
0.
It's 0. Rise over run. No matter how much you run, there's no rise. It's 0 over whatever. So the slope is 0. The reaction rate is 0. 0 mils of oxygen per second means it just is never going to change that. It's going to always stay flat. The other stuff, though, in fact, I need to talk. So I'll have to ask the group that put this data in here, because there's something weird. Trials 1 and 2 seem like they were put in the wrong things, because they don't really make sense. In fact, if let me bring it up. If somebody can, hold on. The person who typed this data in, can
That would be me.
So could you tell me, was this the data that you had for actually for trial 1?
Trial 1. Yes.
And this was the data you had for trial 2?
Yeah.
Okay, so something is off. So this isn't one of these things. We'll talk about that more. So I'm going to stop sharing here.
Are you going to make that available so the rest of the groups can add their data in on Canvas, or?
Yeah,
Yeah.
I am gonna create something. Okay. So let's go back and remember what are the independent and dependent variables for this. So that's going to help us stay focused on what we need to do to analyze the data. What was our independent variable?
Concentration of substrate.
This is the concentration of the substrate. Again, this thing was easy to quantify. It's anywhere from 0 to 100% based on, which trial we're doing. Trial 1 was 20%, trial 2 was 40%, trial 3 was 60%, trial 4 was 80%, trial 5 was 100% H2O2 solution. So this, we got that covered. The dependent variable, what's the dependent variable?
Reaction rate.
It's going to be reaction rate. Right, so ultimately, what we're going to want, if we're going to look for the relationship between these, we're going to have substrate concentration, reaction rate. Substrate concentration, we can just do, again, like I said, just based on the dilutions that we made. Reaction rate was the tricky one. Reaction rate, we said we're going to be using this idea of mils of oxygen per second as a reaction rate. Like how quickly is oxygen showing up? Because oxygen shows up as hydrogen peroxide gets broken down. But that means for each of these trials, this was trial 1. And this is trial 2. This is trial 3, etc. For each of those trials, we're going to have to look at the data, look at the amount of oxygen that's getting made per second. We're going to have to approximate a best fit line, calculate the slope of the line. And that's ultimately going to give us some reaction rate for that trial. So for trial 1, we're going to have one number that is going to be the reaction rate for that trial. For trial 2, we're going to have to make another graph of time versus oxygen in mils and draw it out and draw a best fit line and calculate a slope. And ultimately, that slope is going to be our reaction rate. So for each of these trials, there's going to be this involved process to actually get the reaction rate, which is what we really want. What you were measuring in lab on Tuesday, like the volume of oxygen, that was not what we ultimately care about. What we care about is use that information to figure out the reaction rate on that trial. So is that making sense? The data that you got on Tuesday is just the first step. It's not your dependent variable yet. So what I'm going to do now is kind of go through step by step and kind of talk about how we need to go about actually figuring out that reaction rate. So here, I'm going to just share screen here. First thing you're going to have to make sure, remember, is when you're recording the burette and mill, that's how much water is left in the burette. And what I really care about is how much oxygen is getting made. How do I actually get the amount of oxygen at each time point?
You subtract from the water to start.
Say it again.
You subtract from the 50 and then whatever number it's at.
Yeah, exactly. It's just going to be 50 minus whatever that volume of water is going to be the amount of air that must have displaced it, right? So if there's 50 in the burette, there's no oxygen made yet. If there's 45 in the burette, that means there's 5 oxygen. There's 41 in the burette, that means there's 9 oxygen. It's just 50 minus whatever the burette reading is. So that's going to have to be your first step in this calculation, is get a column of the oxygen volume rather than the burette reading. And then you're going to make a graph. And when you make this graph, is going to be used for calculating actual quantitative values for your reaction rate. So you have to be careful when you make it. So I'm taking a look here. I'm going to make sure, okay, I don't have to go beyond 50 seconds because the reaction's done at 50 seconds. And I don't have to get up higher than 20 because it's 17. So if I make my y-axis go up to like 20 mils of oxygen, I make my time go out to 50, that's going to be just about right to fit all my data here. And make this, I'm taking actually a ruler here on my iPad to try to make this pretty.
For this part, I was reading in the lab notebook that we can either do 6 individual graphs or we can graph them all on one. Is that still correct for options?
It is correct. I'll talk about the pros and cons of the two in a few moments.
Okay, thanks.
So right now, let's just look at trial 1. And I am going to so this is going to be time in seconds. Oh, shoot, wait, 5, 10, 15, 20, 25, 30, 35, 40. This is going to be mils of oxygen.
You skipped the number 30.
Oh, thank you. Holy crap. Okay, thank you. This is going to be, let's see. Actually, this will work. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. So 1, 2, 3, 4, 5, 8, 9, 10. So it is going to be important that you do keep all of your axes well spaced, and definitely don't do what I just did where I had a asymmetrical thing here. Make sure if from here to here is 5, and then the same distance has to be another 5, or else we're in trouble. So then we got to draw our data. I am looking at the data here, so I can just read it. I'll do it here. At 0, it's 0.
11 a.m.
At 5 seconds, it's 0. At 10 seconds, it's 5. At 15 seconds, it's 6.5. At 20 seconds, it's 9. Uh, 25, it's 11. At 30, it's 13.5. Um, 35, it's 16. And then the next one is 17, and it just stays there. So this is what I did. I have my dots. What you need to do is kind of think about what is the broad form of this thing. You know, I'm going to take maybe a green. It's kind of going up, and then it starts kind of flattening out. When you think about where to get the slope, this is kind of the trickiest part here. The slope is not going to be here because that's where we have that weird ass bubble that we are going to, is probably messing us up. We don't want here. Here it's already pooping out. So what we want to do is find this kind of sweet spot, which is probably going to be in the first 30 or 40 seconds, where it's going pretty strong and pretty constant. You have to eyeball it. This is where drawing this manually and eyeballing it is going to be how you do it. Using the computer, the computer doesn't have your sense of what is important or not important. So look, I'm going to take a ruler, and please use a ruler. I mean, sometimes people like, you're going to actually calculate stuff on here. So please use an actual ruler, and you're going to draw your best fit line. Best fit line doesn't mean it goes through everything. It means you are kind of eyeballing what you think is kind of like most likely. So I'm going to say that. You know, there is not a perfect best fit line. There's more than one, you know, depending on your like aesthetic sense, but they should be close. If two or more people drew a line, best fit line, they would be kind of similar. I could imagine drawing this a little steeper, but I think this is probably fine. Notice, does not have to go through origin. A lot of times people think it has to go through the origin. It doesn't. You know, and a lot of times it's not even going through a dot. It's just close to a dot. So this is going to be my best fit line. So here I got my best fit line. So this is the first step. The first step is plot it out in a pretty graph and then draw a best fit line that you think best approximates the slope when the reaction was really going at its best, before it started pooping out here and kind of ignoring the craziness at the very beginning. What's the next step?
Figure out your slope.
You have to calculate the slope. And to calculate the slope, you just have to pick any 2 points. Doesn't matter. Taking any XY pairs will give you the same slope. So I usually, just to make my life easy, I usually take 2 dots that are 10 apart from each other, because then the denominator is super simple. So I am going to, let's go to 20 and 10. Oops, what am I doing there? So here's my delta Y, here's my delta X. Again, you can pick any 2 points. I picked the seconds at 10 and 20 just because when I do the math, it's going to be easy. Now I need to find what is the values. So the delta X is going to be easy. So this is so delta Y over delta X, this is my slope. So this is going to be whatever value the Y is here. So I have to find out, and I look at the line. I don't care about the dot. The line might be on the dot or it might not be on the dot. Don't use the dot or your data as the value. Use the line. And I say, okay, let me come across. And that's around 6, 7, 8, 8.5. So it's going to be 8.5 minus whatever this value is. And again, it's the intersection of the line, not of the dot. When I come across, and it's like 4. No, 3. It's 3. So 8.4, 8.5 minus 3. The delta X, by definition, is going to be 20 minus 10. So it's 10. So this is now going to equal 5.5 over 10. Well, I wish I didn't make this so messy. And that's going to be 0.55 mils of O2 per second. That's my delta Y over delta X. This is going to be my reaction rate for trial 1, 20%. So you're going to have to do this same thing for trial 1, for trial 2, for trial 3, for trial 4, for trial 5. By the time you do it for trial 5, you're probably getting pretty good at it. You know, for each of those times you calculate, you are going to get the reaction rate for that trial. And then you'll be able to take that and you'll finally have your reaction rate. You know, so if this was, you know, substrate concentration, 20%, 40%, 60%, 80%, blah, 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 I'd say, oh, for 20%, it's 0.55. There's my dot for my graph. Now what I'm gonna have to do is get my data for trial 2 and put it on a graph and draw a best fit line for trial 2 data, and then calculate the delta Y over delta X for trial 2, and finally get a number that's a reaction rate for that. And then I can figure out, where does that dot go here?
Where are you getting the 20% for the trial 1? Isn't it like 1 milliliter? So wouldn't it be 10%?
1 mil out of how many?
Out of a total of 5?
Yeah.
So that's 10, right? Oh no, that's 20, right? I don't know.
If you do know, if you think about it, right? 1/5. 1/5 is not 0.1. What is 1/5? What does 0.1 mean?
0.20, I'm sorry.
Yeah. 'Cause
Sorry.
0.1 means 1/10. So this is 1/5. If I double this, would be 2/10, which is 0.2. Yeah, so 1 out of 5 would be 20%. And this one, the one that has 2 mils out of 5, would be 40%, for instance. So that's how we're doing that. So trial 1, trial 2, trial 3, trial 4, trial 5 is going to be just the pure 5 mils of just the hydrogen peroxide. As the question someone asked a little earlier about doing them all on the same graph, you can. A nice thing about doing that is you can actually kind of see them, compare them, and see how they look compared to each other. Like, for instance, I'm going to share part of the reason I know something is weird in this data that I was printing from the earlier group. This is actually helping me realize, like, what's up here. Trial 1 should have less hydrogen peroxide, so by the end it's going to make less oxygen. Trial 2 has more hydrogen peroxide, so by the end it should have more hydrogen, more oxygen. But yet I'm seeing the opposite. I see trial 2 died and had less oxygen by the end, and trial 1 had more oxygen by the end. So that's making me, again, doing my sanity check, something's wrong, right? There's some reason I can't trust this data. My 2 options that I think of, one possibility is trial 2, there was a leak. So we didn't capture all the oxygen. Some of it was just, somebody didn't seal the cork. You have to make it wet and really squish it in. And if you don't, there's pressure building up and gas leaks out. So we just didn't capture all of it. So that's a good possibility, is that we just didn't capture all of the oxygen in trial trial 2. And that's why trial 2 looks so kind of anemic compared to trial 1. Other thing is we can kind of start looking at slopes. Here we can see this trial, this is definitely steeper than this one. So I can say like, oh, that's nice. It's kind of nice to see even just ballparking it. Higher concentrations were going faster than the lower concentrations, was what I expected. Whether or not we're going to actually see saturation, you're going to have to do more of a fine-tuned analysis of what are the slopes, and we won't know that until after the group looks at it in more detail. But so you can do them all on the same one, but it gets tricky because of they're on top of each other. You have to make sure you color code things really well. You make it sure it's as big a graph as possible on a whole page so it's not squished in. So I find it's often just less confusing to just do each one on its own little graph. That way you don't get confused about which dot belongs to which trial. But both ways work. Or you can even do a combination.
Do we need to be like connecting the dots and then doing the best fit line, or can we just put the dots in and then do our best fit line?
Yeah, no, I only connected the dots here just to make it look kind of interesting for right now. But yeah, no, you don't really connect the dots.
We just do our best fit line.
Yes.
Okay.
What I do sometimes, though, as I was kind of showing you, because sometimes, here, let me, yeah, sometimes like here, which one? This blue one, trial 5. If I'm going to try to get rid of everything but trial 5, give me 1 second here. What? Cancel, but I did the opposite of what I meant to do. Okay, here is trial 5. And it's kind of, in fact, let me get rid of just make it a more classic scatter plot. Okay, that's more what you're going to be drawing. Something that looks like that. And what I find is sometimes I don't even know if it's like, it's not necessarily a classic way to do it, but I find it's useful for me, is I kind of do try to draw like a broad line of like, these dots are not really, there's a lot of wiggle up and down because those bubbles are weird, right? Sometimes the bubble is still coming up when you're ready to try to read it, or glommed on. So I would say each of these dots could be higher or lower than it really is supposed to be, because those big bubbles were making things really awkward. And so I try to think about where is the actual real curve underneath the wiggle of the dots going up and down. And then I can like, say, okay, it's probably more. So then draw my line. It's kind of like that. But it kind of helps me to draw this big thing in like that to kind of get a sense of, oh yeah, up in this part, it's already, I'm kind of making it harder. Up here, it's clear, it's already starting to kind of poop out and no longer be at its kind of main slope. So I find as you look at the dots, you don't want to connect them, but you do want to try to intuit, like what is the true graph that these dots represent, given that there's messiness into how the dots are actually being measured, right? Because, ideally, we'd have like finer little bubbles rather than big gloopy bubbles. And we've been trying to play with that a little bit, but we didn't do it really this time. So does that, I don't know if that makes it clearer. And again, if you've been, I think Chem 114, you've done this already, but in like Chem 110, it's basically tries to like do everything in a super inadequate amount of time. So people come out of Chem 110 usually kind of confused about chemistry rather than knowing everything they need to know. So this is what, so for this is trial 5. Again, if I'm gonna get the slope, I can just pick any points. Let's pick the 10-second one and the 20-second one. So I can go, here's a 20-second from here to here. That's going to be my delta X. This is going to be my delta Y. And I just have to find that intersection of the green line I drew here. And I would say, okay, this is, I'm going to ballpark it. This is around 17 minus 10. So it's about 7. Again, I know this is 10. So this would be like, 0.7 mils of O2 per second. So when I go back to my little graph of substrate concentration versus reaction rate for trial 5, that's going to be my reaction rate. It's going to be the slope I calculated from drawing my best fit line and calculating the slope for trial 5. So does that make sense? I'm going to reiterate what I said last time, just because people still get confused around the idea of enzyme saturation versus just a reaction ending. Because there is something that looks physically similar on the graphs, but what you're looking at is very different. So just to reiterate, when you're looking at a single trial, and you're looking at time in seconds versus mils of oxygen, which is the one you're going to need to do for each of these slope calculations, it's going to go up and eventually it's going to flatten out. It's going up. This is where the reaction is running. Hydrogen peroxide is breaking down, making bubbles. That's why the level of oxygen keeps going up. What happens here? Why is it flat here?
Because the reaction's done.
Reaction is done. All the hydrogen peroxide is broken down. There's no more oxygen being produced because there's no more substrate left. which means you can wait till the cows come home and the mills of oxygen are not going to change anymore because there's no more substrate to break down. So no more oxygen is going to get made. So this versus the graph that you're going to make after you've calculated all the reaction rates that you're going to use to actually check out your hypothesis. So after you're going to get these slopes, delta y over delta y over delta x is going to be the rate. And then you're going to make a second graph, which again, I'm going to substrate concentration versus reaction rate, moles of O2 per second. Again, you're going to calculate the slope. That slope's going to go here. Another trial, you'll calculate the slope. It'll go here. Assuming it very well could end up looking like this. Which has the same physical shape. But what's happening is really different. What does it mean here when we see this slope going up?
Well, it's the reaction, right? Instead of the oxygen, right?
And what is it telling us? What conclusion can I draw from looking at this graph here?
That the higher the concentration of substrate, the faster the reaction rate, or the higher the reaction rate.
Exactly. So this is telling us here that at least in this region, we have a direct correlation between substrate concentration and reaction rate. As we increase the substrate concentration, the reaction rate goes faster. These slopes are getting steeper. What about on this side? Here it's getting flat. What does this flat part mean?
There's no more reaction.
At the saturation?
So we've ended up with enzyme saturation. So is there a reaction happening?
No.
Yes.
Absolutely. Remember this dot, how do you calculate these dots? These dots are the slope. So to get that value here, each of these slopes, each of these is a delta y over a delta x.
So the reaction's happening, but it's like the same.
It's happening, but it's the same rate. The rate is
Same rate, yeah.
Yeah. So that's why I want you to really get this distinction. When you have a flat line up on the top here, the reaction is over. When you have a flat line here, the reaction, by definition, these dots are when the reaction is running strongly. That's when you calculate the slope to get the value to draw that dot. But having this flat just means that we're increasing concentration, but we are no longer increasing reaction rate. Again, it would be like if this was time versus moles of oxygen, here, and then it's like, here I'm going at a certain rate defined by the slope, making a certain amount of oxygen. At this point, this is how much oxygen I've made when the reaction's done. In the red case, we can see it's going at the same slope, means the rate isn't any faster. The rate is the same. It goes longer. By the time you're done, you've made more oxygen because you had more substrate to start. But the rate is the same. So like if I maybe I'd have, if I was looking at this, a graph like this, the yellow dot would be like, here, the blue dot here, because it's steeper, right? Yellow, blue is steeper on blue, but then blue, red, and green are all the same. So in this case, we can see this idea that it's going up and then it flattens out. Again, this is concentration versus rate. The rate is isn't getting faster, but it doesn't mean that the reaction isn't going. The reaction's going crazy. So that's, again, and I know people get confused here. That's why I'm going over it again and again. I do understand that it can get tricky to keep this all straight. So are there questions about that? Okay, so what I would like you to do then, like, over the weekend, do those, try to get this, not this, but try to get this graph. I want you to get your first attempt to get this graph of the independent versus dependent variable. To make this, you're going to have to make 6 separate graphs to get each of the data points. Right? To make this graph, you have to make 6 of these graphs. I also would like, I think the easiest thing is have one person from each group just email me like a picture of your data sheet, and I'll put stuff in together. And so after everybody like analyzes their individual data, we'll be able to kind of average things together and see if that cleans things up a bit, which it might. So are there any questions about how you analyze this? Like when I'm grading this one, you don't get as many points for just being, writing down the numbers as actually doing the analysis. It's important if you want to, get credit for this lab that you understand how to actually analyze it. How do we actually get the reaction rates, that we can then use to check if our hypothesis is supported or not. So if you're kind of lost in this, you can reach out and you can ask me. I'll try my best to clarify it. Or maybe you're going to start it and then hit a wall and then send me an email or something. But make sure that you try it. And if it gets confusing, that you reach out and get clarification. All right. So any last questions, comments about Enzyme Lab before we roll on? Okay. Maybe let's do another 15-minute break, and then we're going to talk about the literature. So let's come back at 11:45 a.m. and talk about all the interesting, cool studies that are out there for you to explore.